So good afternoon, everyone. Thank you for joining me today. My name is Weining Lin from University College London, and I'm very sorry that I cannot present to you on site due to the visa issue. But it is a great pleasure to introduce my research on the protein function prediction model that I have developed. And this model, we try to leverage the power of the ensemble learning to provide a more accurate and comprehensive understanding of the protein functions by combining the information from three most important aspects: the sequence, the structure, and also the interaction networks. So as many of you may already know, all organisms rely on the correct functioning of their cellular workhorses, which is protein, which are involved in nearly all roles. So from, for example, catalyzing chemical reactions as the enzymes to acting as the transmitters through the cellular signal transduction. As so many fundamental functions are carried out by the proteins, so understanding the protein function is critical and essential, especially for the advancements in the fields such as the drug discovery, disease understanding, and the synthetic biology. Even though the interplay between proteins are very complex, the gene ontology tries to capture this complexity and also to try to standardize the vocabulary used to describe the protein function in a human and machine readable manner. The gene ontology separates different aspects of protein function into three categories, which is the molecular function, cellular components, and the biological process. And to be more specific, the molecular function is like what activity of the protein at the molecular level does it carry out. And the cellular components is where exactly within the cell does this activity occur. And the last one, the biological process, is what larger biological process does this activity contribute to? So due to the recent development in the high throughput sequencing technology, the number of protein sequences increased dramatically. And I checked one of the most important protein sequence database, which is UniProt, and I noticed that there are around 250 million protein sequences. However, there's only around 1% of them have the annotations verified by the expert. And this is mostly due to the fact that experimental determination of the complete gold terms is quite challenging. Thus, I can say that the gap between the number of proteins with the experimentally verified gold numbers and those with known sequence but unknown function remains substantial. So in the past few decades, many computational methods have been developed to try to bridge this sequence annotation gap. So in most cases, this method based on three different protein features. One is the sequence similarity level based method, which basically they try to transfer the annotation from one protein with a known function annotation to others that without any annotation, if the sequence they are similar enough. Because for the proteins with a similar sequence, they usually share a similar function. And that is also true for the structure-based method, which uses the three-dimensional conformation of the proteins to infer their function. Because the protein with a similar structure, they tend to perform a similar role. And for the last one, the protein-protein interaction-based prediction. So the biological principle behind this method basically is for a protein, if they have the interaction in the same biological pathway, their partner often share a functional similarity with this protein. However, most methods only use just one modality. As these features are so important in determining the protein function, can we combine together? And what if we combine together? So to explore this idea, I attempt to train models individually for each modality. And for each part, I try to use the most suitable protein language model to perform the visualization, ensuring that the unique characteristics of the sequence, the structure, and also the interaction data can be effectively captured. So let's start from the first modality. So in order to leverage the evolutionary information, I constructed multiple sequence alignments for each target sequence by finding their homology sequences. Then we feed these MSAs into the MSA transformer. And this protein language model was trained on 26 million MSAs. So we can say that the MSA extracted from the MSA transformer contains very rich evolutionary information, which is critical for the accurate protein function prediction. So once we obtained such evolutionary rich embedding from the MSA transformer, we feed them into a nonlinear multi-layer perceptron and trained the model to predict the protein function based on these features. And for the second modality, even though in this part, we call it a structure-based model, but the input to the model is just a sequence because we leveraged a recently released protein language model called the ProstGOPred. And this model is quite unique because it was trained simultaneously on both the sequence and the structure. So to be more specific, the ProstGOPred encodes a protein structure as a token sequences using the 3DI alphabet introduced by POSIX. And the model was trained to translate between the 3DI sequence and also the amino acid sequence. So this innovative training strategy allows the generated structure sequence representation can effectively capture the complex relationship between the protein sequence with its structure. Therefore, such structure we're embedding, we get from the ProstGOPred. We believe that it should theoretically highly effective for predicting the protein function. So after we get the embedding from the ProstGOPred, we feed it into a nonlinear layer to refine the feature. But make the further prediction, we applied a contrastive learning strategy. So to be more specific, we vectorized the Galton label into representation. So in each batch, a protein, it does not only serve as the anchor, but also serve as a positive sample. So that means any other protein in the same batch, they are all considered as negative samples. I know this strategy may sound a bit strange, but it proves that it works. We compared the result of this strategy with with the other method based on the binary cross entropy, and this method have a much better performance. And we believe that this effectiveness likely arises from we align the protein feature representation with a label representation in the latent space, which facilitates easier and more accurate learning for the model. And for the last one, to leverage a protein interaction network information, we constructed a protein-protein interaction graph using the data from the strain database. So the strain database, it provides a confidence score for each interaction pair, which is a way to measure the reliability of the protein-protein interaction. By utilizing these confidence scores, we were able to establish a comprehensive protein-protein interaction graph. And each graph was constructed by selecting the top 100 neighboring proteins for each target protein based on their confidence score. And so basically that's it. And as I just mentioned, each node is a protein, and each protein's sequence, we feed it into the ProstGOPred. And then we take the embedding for each protein as the node feature. And then we feed the entire graph into a graph neural network for the further process. So that is, we applied an attention-based neural network to assist the interaction processing. And in the GAT layer, each node embedding was computed by attending to its neighboring nodes, so that each node can aggregate information from the neighbors using the attention mechanism. And using this, so in this case, during the training, the updated embedding can incorporate information from their respective neighbors. And then we conduct the global average pooling of the updated embedding to create a 1D graph representation. And then we feed the representation into the feed-forward neural network for the final prediction. So these are the optimal strategies we develop for each of the most critical modality in protein function. And for our method, we just simply to apply the ensemble learning by integrating these methods together. And we assign equal weights to the prediction outputs of each model and then perform a weighted sum to obtain the final prediction. And I believe some of you may ask, have you ever tried any ways that optimize for each subontology? And the answer is I tried. And surprisingly, I found that using the equal weight has the best performance. That is pretty surprising and interesting to me. Could be a bug, but could also be some interesting phenomenon behind it, which I will give some further investigation in the future. But anyway, with this manner, our approach can leverage the strengths of each individual model, which can therefore give a more accurate final prediction. So when we compare the results of the models trained on each individual modality at the kappa-3 dataset, we notice that the structure-based method gives the best performance comparing to the MSA-based method and also the interaction-based method. But compared with the ensemble method, we can see that the ensemble method significantly improved the performance in all subontologies. Therefore, it is obvious that using the ensemble method can indeed enhance the performance in predicting the protein function prediction task. And we benchmarked our method on the structure-based benchmark with other SOTA method, which were all trained on the 3D conformation of the protein structure. And we noticed that even without using any protein structure in the 3D conformation, our method is very competitive with this method. So in biological process and the molecular function subontologies, the performance of GoBeacon is very close to the SOTA method, while the GoBeacon's performance is even the best one in predicting the functions under the cellular components category. And we also benchmark our method with other SOTA methods on the kappa-3 dataset. And our method has a bit improvement, which result in our method as the best one now in the kappa-3. But I really want to mention here is most published method only use just one modality. But in the kappa-5 competition, we can also notice that more and more people, they are trying to explore to predict the functions using the ensemble method as well. So even though GoBeacon achieves the best performance on the kappa-3 benchmark and very competitive with other structure-based models, there are still some space that we can improve our model's performance. For example, recently, many studies prove that including the Galton textual information can improve the model's performance. Some study apply the synthetic similarity of the Galton as a feature during the model training, while some study took the embedding from a large language model trained on the literature. Either way, they are sounds very interesting to me and worth to give a try. Besides, the contrastive learning we apply in this study is quite simple and naive. However, since the Go graph is in a hierarchical manner, we did not really include the relationship between Go terms during the model training. Therefore, in the future, we'll also try to train the model with the contrastive learning, considered the hierarchical information of the Go tree. And for the last one, I believe many of you may already have heard about that, is the release of the latest ISMB model. And this model was not just trained on the sequence and the structure, but also include the protein function information. So I'm quite looking forward to test our method on using the embedding extracted from the ISMB. And I would just say thank you for your attention, and thank you to everyone from Orango Lab and also our collaborator, especially David. Thank you so much for your help.
Thank you. Questions?
In your first model, okay, in your first model where you use ESM MSA, the MSA was generated by HHBlitz, which is optimized for structure prediction purpose only. So have you actually compared ESM MSA and ESM2, for example, for the same purpose?
Yes, I tried. And ESM2 has actually a better performance in the MSA. But when we combined the model trained on the ESM2 with other models, the model's performance dropped a bit. So we believe using the ESM MSA models embedding for the sequence-based method, it can actually has the complementary effect with other modality.
Okay.
Other questions? Maybe I can ask one question then. So in our lab, we are also using ProstGOPred, but PTM site prediction. And it looks like for site prediction, ProstGOPred is not as good compared to protein level prediction. Do you have any observations like that?
Sorry, I didn't really hear you clearly. Can you somehow repeat your question again?
My question is, you used it for protein prediction, right? Protein level prediction.
Protein level prediction, yes.
Yeah. Have you any insights on site level prediction for ProstGOPred? Its performance on site level prediction.
What do you mean by side effects prediction?
For example, if you're predicting a binding site in a given protein or something.
No, I haven't tried that yet. But I have another project, and we are going to test we call it druggable project is the test the important binding residue of the given protein for the ligand. I assume you would like to ask something similar with that one, but that is something we are going to test in the future.
Okay, sounds good. Thank you.